knowing that there's a patient out there, and to know that something that you worked on every day is now out there in the world, making an impact, saving lives.
There is so much innovation being made. I haven't felt this level of excitement and this level of interest in the field since the Human Genome Project. We know how fast the field is developing, that there is competition to get to market first. We have become experts in the field by working on some extremely large molecules from the onset.
We want to be an inspiration. We want to be a difference maker. We want to be on that cutting edge of science. We're a GMP facility for the manufacture of drug substance.
We have the two platforms. It's our CHO-based monoclonal antibody platform for therapeutics, and then our newly rolled out messenger RNA program.
We are going to be able to offer you a full array of services. We're able to offer it all in-house.
We provide upstream cell culture and downstream purification manufacturing operations, as well as process development and analytical development.
From monoclonal to antibodies, we've built a platform here at Curia that we believe can usher a client from construct to clinic in 12 months. It's a very aggressive timeline, and that's perhaps why you don't see it in the industry.
That includes developing the DNA platform, going all the way through the cell line selection, developing a master cell bank, scaling up the process, optimizing the process to get you into your GMP production run up to 500 liters.
We've built a general scheme, and we've created menus against each of the major unit operations with kind of some predefined choices. And we've built inventory and batch records against those predefined choices.
So we were able to achieve a 12-month timeline from early discovery all the way to GMP manufacturing by trying to eliminate as many variables in advance of entering the production space. Alignment of materials that are being used, alignment of release assays that need to be run, and coordinating activities across multiple sites.
Unifying procedures, practices, SOPs, equipment, from again that early bench scale operation all the way through our GMP manufacturing operations.
Our development scientists are working right alongside our analytical development scientists as well as our GMP level operations scientists.
We built our GMP team perhaps a little differently than maybe most have. We felt that the better way to serve our clients, a better way to operate was to have subject matter experts on the floor executing those operations. It was out of some of our investigations into advanced therapies beyond monoclonal antibodies. We began to see that we could translate some of the great skills here and some of our great team to an mRNA platform as well.
At Curia, we started our innovative science focusing on a self-amplifying RNA molecule, which is leaps and bounds above the potentials of some of the traditional mRNAs that are currently being used in therapies. Most RNA molecules that have been used in the field are relatively small in terms of size, from 1,000 base pairs to 4,000 base pairs. By focusing on the self-amplifying, we've been able to handle ones from 9 all the way up through 16, and we're pushing into 20 kb, which so far we've seen no one else be able to truly make, optimize, and be able to put out into market. With the process we've developed, we've established that we can get from start to finish into phase 1 studies within 6 months of inception of the program.
The analytical development team is in charge of developing the assays for the GMP run.
There are compendial assays as they're related to the USP pharmacopoeia and the European standard. And those are your basic sterility, bioburden, pH, osmolality, concentration, where we go by a specific protocol that's set out in the USP and guidelines for testing. We do a verification of those. Many of the other residual assays and purity assays are going to be customized, and that's part of the process of analytical development, is to take in the material and run that through the test and see where we need to go with it. Does it meet the specifications and criteria set out in the assay? The other part of QC that you don't really see is the environmental monitoring that goes on that's attached to our GMP suites.
You need to make sure that the environment in which the material is being made is free from any potential microbial contamination. If you're looking at biologics, there's no terminal sterilization, which means the process has to be clean from the beginning to the end to minimize any potential contamination. We trend all the environmental monitoring data from the clean rooms. Right at the beginning of every project, my goal is to see what the molecule is, what the material is, and is it compatible with those assays? And if it is not, how can we make adjustments to make sure that we can test it the right way when the time comes to perform it under GMP.
Now, obviously, there's a number of mRNA therapeutics in the market right now, but the roadmap for analytics is still under construction. What's happening now is that we're right on the front edge of it. and I get to sign off on releasing drug substance and drug product and know that we did fantastic testing, know the molecule's good, know that it's safe, and confident in the fact that we performed a really, really good process and we're helping these clients push things into the clinic.
We care about getting these life-saving products to market. We care about our customers effectively bringing products to their patients.
You want to have a sense of accomplishment that you were part of a team that was able to bring life-saving medications to the world.